So here we go to the next page, and here we see T cells and B cell activations. So antigen presenting cells are B cells, dendritic cells, Langerhans cells, and macrophage. Now two signals are required for T cell activations, B cell activations, and class switching. Now talking about T cells activations, the very first thing we can remember, that is antigen presenting cells ingest. Here we have antigen presenting cells ingest and process antigens. So antigen presenting cells, that is either dendritic cells, B cells, Langerhans cells, or macrophage, that ingest and process antigens, right, and then migrate to the draining lymph node. Now second thing, that is activations of signal first. T cell activations starts here, and signal first is like exogenous antigen is presented on MHC class 2. So if antigens is exogenous, so exogenous antigen is presented on MHC class 2, right? So this antigen coming or engulfed by endocytosis by antigen presenting cells, if it is exogenous, then it is presented by MHC class 2 and recognized by TCR, that is T cells receptors on T helper cells, that is T CD4 positive cells, right? So if antigen presenting cells is presented by MHC class 2 and it is exogenous antigens, then this must be uh, this must be T helper cells, that is CD4 positive, right? Now endogenous or endogenous or cross presented antigens is presented by MHC class 1. Similarly, this antigen, if it is endogenous antigens, then this antigen presenting cells will present it through MHC class 1 to T cytotoxic T cells. This must be cytotoxic T cells if it is presented MHC class 1. Now this up to this signal first is completed. Now moving up to the signal second, proliferations and survival signals. Okay, it is signal 2. So in this case, costimulatory signals through interactions of B7s, that is proteins, B7's proteins, that is CD80 and 86 on dendritic cells and CD28 on naive T cells. So in second signaling, after presented by antigen presenting cells, and it is bind by connected to naive T cells, or we can say according to the MSC, if it is MSC class 2, then it is connected to T helper cells, and if it is class 1, then it is connected to MSC that is cytotoxic T cells. So up to that signal first is finished, and now for second signaling, this antigen presenting cells again connected to these T cells with these B7 proteins, that is CD18 or 186 with CD28. Okay, now activated T helper cells to produce cytokines. So now after this second signaling, these T helper cells produce cytokines, and T cytotoxic T cells able to recognize and kill virus infected cells. So after completing these two signaling, this either if it is cytotoxic T cells then started to recognize and kill virus infected cells, and if it is T helper cells, then it produces cytokines. Now for B cells activations, we can see here that T cells activations is above, as we have seen above. T helper cell, after T helper cells activations, okay, B cells receptors mediated endocytosis. So here we can see B cells receptors mediated endocytosis. That is, here we have B cells, and it have receptors, that is B cells receptors. So antigens is endocytosed in these cells, and then exogenous antigens is presented by MSC class 2. So if it is endogenous antigens, so it is presented by MSC class 2, which is uh, bind to CD4, that is T helper cells of this activated T cells. Activated, that is CD4 T helper cells, right? And recognized by TCR, that is T cells receptors on T helper cells. So we already discussed about this. Now second signaling is like CD14 receptors on B cells. So here CD14 receptors on B cells binds to CD14 ligand receptors on means ligands on T helper cells. So this is second signaling, and then the cells now secretes cytokines. This CD4 T helper cells secretes cytokines now, and then determine Ig class switchings of B cells. Now this further determines Ig class switchings of B cells. Now B cells are activated and produced IgM. They undergo class switchings and affinity of maturations. So this much for now.